Good morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Allison Baksh, Marketing Manager at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Robust Detection of Translocations and FFPE Biopsies with TLA-based NGS, Application in Lymphoma Diagnostics. This webinar is sponsored by Surgentis. Our speakers today are Daphne de Jong, pathologist at Amsterdam University Medical Center, Dr. Bauke Ylstra, Director of Tumor Genome Analysis Core at the Amsterdam University Medical Center, and Dr. Harma Fietsma, Head of Business Development Oncology at Surgentis. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look at the bottom tray of your window, you will find a series of widgets to enhance your webinar experience. Okay, with that, let me turn it over to Dr. Daphne de Jong for her presentation. Please go ahead.
Thank you, Alice, for that introduction. I would like to start out this webinar by giving some background on the role of translocations in malignant lymphoma, why this information is important for pathologists and hematologists, and I would like to discuss the current practice on how we demonstrate translocations, so current daily practice. Well, malignant is a very large collection of diseases, almost 100 different classes and subclasses of B and T cell lymphomas, that are all defined by a characteristic morphological and immunophenotypical spectrum, by a characteristic genotypic set of features and a typical clinical context. These diseases range in aggressiveness. Well, genetic alterations in malignant lymphoma are part of the classification, especially as it holds true for translocations and mutations. Many mutations can be found in lymphomas, follicular lymphoma, characterized by 14/18 translocation with BCL2 rearrangements, but also mutations such as the very well-known MID88 mutation that is so characteristic for lymphoplasmacytic lymphoma. They are characteristic but not specific. This is the classical way to approach the use of genetic alterations. Increasingly, we use genetic alterations also for prognostic stratification, especially in diffuse large B-cell lymphoma. We use gene expression profiling, we use next-generation sequencing profiles, and also sets of translocations. Predictive information to guide treatment choices is finding its place in lymphoma diagnostics and treatment. So of all these genetic alterations, translocations are currently the best defined. These are often very early alterations and therefore not only diagnostic, but can also be used as clonal markers, for instance, during follow-up as minimal residual disease markers. Translocations lead to direct or indirect deregulation, often direct deregulation, of oncogenes. Again, follicular lymphoma with its 14/18 translocation, its BCL2 overexpression that leads to its oncogenetic effect. Most translocations are reciprocal, meaning that a piece of chromosome A is transferred to chromosome B and vice versa. And thereby it leads to the overexpression as the oncogene comes under the regulation of a driver gene. How do we do that in daily practice then? Well, when a translocation leads to this specific overexpression at the protein level, the most simple way to demonstrate the translocation is indirect, by immunohistochemistry. And so as an example, on the left side of this slide, you see a mantle cell lymphoma one of the many B-cell lymphomas that we have that is characterized by a 11-14 translocation, translocating the cyclin D1 gene to immunoglobulin heavy chain gene, leading to overexpression of cyclin D1. As cyclin D1 is only expressed at very low levels at all other cells in the lymph node, but the tumor cells, we can very easily see that overexpression of the oncogene in the lymphoma, and that is very helpful for our diagnosis. Similarly, on the right-hand side, you see an example of anaplastic large cell lymphoma with ALK1 translocation, and in this case, the immunohistochemistry even gives us a clue to the translocation partner, showing either a combined nuclear and cytoplasmic expression, as you see in the left-hand panel, or a cytoplasmic expression pattern only on the right-hand side that is dependent on the translocation partner. Well, unfortunately, overexpression is not always uniquely mediated by translocation, and in most cases, protein overexpression does not equate to the presence of the translocation. And therefore then, we cannot use that as a surrogate for formal demonstration of the translocation. This is, for instance, the case in the MYC gene that we will be discussing extensively later. So in practice, we cannot avoid using formal translocation detection, and we do that using fluorescent in situ hybridization as the most widely used method. And mostly we use so-called break-apart assays. That means that the probes, the fluorescent probes, are located on either side of the breakpoint. And in the lower panel, you see probe 1 and probe 2, the red one and the green one, on either side of the region, the chromosomal region, where breaks take place. With a break, the two probes separate, and we see that as single red and single green signals. If there's no break, we see a combined signal in the combined color red and green or yellow. Three examples here: a MYC break, a BCL2 break, and a BCL6 break. And as you see, we do not see the difference, and we really need to look at the label on our slides and carefully look at the label on the file, because the patterns will be the same in the fluorescent picture. Well, alternatively, we can use fusion assays, not break-apart assays, and in that case, the probes are located on the two translocation partners. Probe 1 on one side, for instance on the oncogene, and probe 2 on the other gene, the driver gene. I've used the MYC, BCL2 and BCL6 as examples here because they have received most attention recently, also by our clinical colleagues. And that is because there is a prognostic impact of these three genes, these three translocations in diffuse large B-cell lymphoma. So what we know largely about the situation of MYC in diffuse large B-cell lymphoma, based on quite a lot of studies, is that MYC rearrangements, MYC translocation, occurs in 10 to 15% of diffuse large B-cell lymphoma. It is associated with inferior outcome, both progression-free survival and overall survival. However, the prognostic impact of single MYC rearrangement without other rearrangements is not known, not clear, as is the combined MYC BCL2 BCL6 translocation status. Similarly, also the impact of the translocation partner to MYC, either immunoglobulin or non-immunoglobulin partners, is unclear. So therefore the Lymphoma Lymphoma Biomarker Consortium started out this large study, starting out with more than 5,000 diffuse large B-cell lymphoma patients uniformly treated with R-CHOP. We ended up having almost 2,400 patients with complete clinical information, immunohistochemical information, and FISH information. Those split out, about 11% had a MIC translocation, and those again split out per IG, non-IG partner, and MIC as a single alteration, MIC single hit, versus MIC double and triple hit in combination with BCL6, BCL2, or both. The results, we think, are very important, as these show that actually MIC rearranged diffuse large B-cell lymphoma has a similar survival as DLBCL without a MIC translocation, the red curve in this graph. And only the double hit and triple hit cases actually show the poor survival that is reported. And even that, it is restricted to the first 2 years after diagnosis, while after the first 2 years, survival is similar to the other two curves, and there is no additional impact of the translocation status. And this holds true for both progression-free survival and overall survival. I'll this down more by translocation partner. And it shows that, and this is the green curve, both for progression-free survival and overall survival, that it's the double hit, triple hit diffuse large B-cell lymphoma with an IG translocation partner to MIC that actually have this poor survival, while all the other translocation contexts have a similar survival that does not differ from those DLBCL that do not have these translocations. So in summary, it means that the problem is in diffuse large B-cell lymphoma with a double hit, triple hit context with an IG partner. And that there is a rationale to treat these patients differently, while there is no justification yet, or no justification to treat patients that do not have this specific context any different than we do our regular DLBCL patients using our CHOP. It means that pathologists should determine this detailed translocation status. And that, as said, we routinely do using FISH, fluorescent in situ hybridization. That has advantages because it's suitable for our routine formalin-fixed paraffin-embedded material. It's widely available with commercial probe sets and assays readily available. We can implement it in our normal automated workflow in parallel to immunohistochemistry using the same machines. And we have the correlation to morphology that obviously pathologists like very much. There also are disadvantages because we depend on good morphology. And scoring is very labor intensive and also often subjective and difficult. It's dependent on the probe localization, and with a limited set of probes available, we only have very limited information on the translocation partners. Let me go in detail on this. The left-hand panel shows very beautiful morphology of a diffuse large B-cell lymphoma. And you can imagine that in this case, it's not so difficult to discern the red, green, and yellow dots. But at the right side, the nuclei are smeared and damaged, and it is exceedingly difficult, if not impossible, to actually appreciate those fluorescent signals. Moreover, we often get those very tiny biopsies, needle biopsies, endoscopic biopsies, often necrotic, that even makes it more difficult to be able to perform these assays. Probe localization, and a technical aspect. When I first showed you how we choose the probes on either side of the breakpoint, that sounds quite easy. And in the top panel, you see the MIC gene. MIC is that little black dot in the middle. It's a small gene. But the breakpoints are located at a very wide chromosomal area around the gene. Now, if those breaks fall between the red and the green probes that you see in the top panel, called physis, then we see a yellow untranslocated allele and a red and a green dot of the translocated allele. However, when the break falls within one of the probes, and when the probes are not so super localized, such as in the lower panel, and fall, for instance, at the right, within the right probe, you see at the bottom panel only a single red signal that is separated off, while the other half of the probe, let's say, gets associated with the green probe still and gives another yellow signal. If the breakpoint falls way out of the localization of the probes, we just see two yellow signals at the far right, at the far left side. So it's not as easy as it seems, and the upper choice of probes is better than the lower one, but still not optimal. Similarly, when we use fusion probes, where one single probe is located on the one gene and the other on the translocation partner, the patterns get very complex, as you see in the lower examples. So is this a problem? What can we see and what can we not see? Well, based on our Lunenburg series and also other larger series, we know that almost 40–50% of the MYC translocations have a Ig heavy chain partner. We have fusion probes for that, and we should be able to detect these. 5–10% have an Ig light chain gene partner. There's no commercial probes available for that site, so we cannot assess those translocations. And about 50% have a non-Ig partners, and those we cannot positively identify because we do not know the partner and we cannot use a fusion assay. So that is also a limitation to this technique. So what we can say is that there is room for improvement. And if we would improve and go to another method of translocation detection, that method should be able to provide information on both the oncogene break and the translocation partner. Should be applicable to formalin-fixed paraffin-embedded material, should be robust and reliable also in suboptimal quality material and in small biopsy samples. Obviously well reproducible, preferably quantifiable so that we can say something about clonal and subclonal events, should be widely available to all, preferably as an in-house assay to be performed within a pathology lab, and also a very important one, should be affordable. And if we look at a broader diagnostic need and to future clinical need, we should look beyond translocation and also be able to look at mutations and copy number alterations, such as amplifications and deletions, because those will play increasingly a role in diagnosis and eventually will be important for our clinical colleagues to support their treatment choices. With that, I would like to finish my presentation and hand over to my colleague, Bauke Ylstra. Thank you.
Yeah, so hello, my name is Bauke Ylstra. I'm a colleague of Daphne in the pathology department of the Amsterdam UMC, and I'm a professor in tumor genome analysis, so I'm much more on the research side. And yeah, I think Professor de Jong, diagnostic routine, she needs a robust translocation detection from FFPE materials. So she came to me as I've built much of my career in the detection of copy number aberrations from FFPE material, and later mutation detection, and together we embarked on a combined detection of translocations with the mutations and the copy numbers using next generation sequencing. Yeah, I will guide you through this endeavor and end my presentation comparing our NGS capture procedure with FISH and with the Surgentis TLA method. Yeah, so for us in research, FFPE materials are of particular interest since there are large archives of FFPE tumor materials worldwide. This is actually the archive on the left of the Amsterdam UMC. And the neat thing of those archives is that they have extensive clinical follow-up. So very cool to do research with. So from the FFPE blocks, we take sections that are done evaluated by a pathologist like Daphne, and thereby we can also delineate or demarcate and enrich for the malignant cells if necessary. And then in the laboratory, we can just like use a scalpel and scrape the region that has the highest tumor cell percentage. For lymphoma, this enrichment step is often not necessary since the material is already very rich in malignant cells, and we often use just scrolls for this. Yeah. So DNA that is then isolated from the FFPE materials is very often compromised. So the whole paraffinization and fixation is all not made. It's made to have a good morphology, but it's not made to have good DNA. So we have a lot of crosslinks, base pair transitions, and most of all, the DNA is very fragmented. So because of a lot of breaks in the DNA during the fixation process. Well, we took this DNA isolated from the FFPE materials to do copy number analyses. And what we did, we took the DNA and simply performed whole genome sequencing with this DNA. And then we mapped the reads against the human reference genome, all the reads that we have, let's say 10 million reads or something, and then we counted per position. We made bins across the genome, and we just counted how many reads do we have at which position. And this worked very, very efficient. And meanwhile, we've published this work in 2014, and we've meanwhile processed thousands of samples. And technically, this always works very well. Of course, if like the DNA is really not good enough anymore because of necrosis, you have a worse result. But in the end, like this always works very, very good. I'm going to show you an example because this is just a cartoon, of course. I'm going to show you an example of the DLBCL sample. And here we see the result of such a whole genome sequencing of FFPE isolated material. And I have to point out here, we do not use reference signals. So we use the reference genome on the internet. We just use that to map our sequences to, but we don't use a normal reference. And here we see 84,000 black spots, and every of those black spots is a bin where we've counted the amount of sequence reads. And so from left to right, we see the different chromosomes, and from top to bottom on the left, on the y-axis, there is the gains and losses. And the gains and losses are furthermore in red, and I'm lost here because, oh, here's my arrow, sorry. Very good, let's continue. And so we see on chromosome 2, we see a very high level amplification. So we have a very small region where we have many copies of, and then on chromosome, 5 and 6, we have gains, 6p has gains, and then on 6q, we see losses, and then further more down the line from chromosome 13 on, everything is quite normal. So genome-wide, we have a very, very high resolution of gains and losses from this FFPE material, and again, we've done thousands of those samples. So now we got a flow from the scrolls or the scrapes or the FFPE cores to this copy number analysis workflow, and we do a single read 50 for this. So there's not much done with the library, but this same library, we can now say, okay, we are interested also in mutations of a set of genes, and what we can do, we can use that same library and do a sequence capture. So we capture those genes that we are interested in, and we sequence that with a paired end approach. In this case, like paired end 125. So from both ends of the fragmented DNA, from both ends we sequence. And then we get sequence reads from this, and those sequence reads we can also map back to the reference genome, and then you look in the reference genome, so all those gray little lines that you see here from top to bottom, all those gray little lines there, each of them is a separate sequence read, so we really have like a read depth here for the captured regions of about 100 to 500 deep or something. And then we see this, that for this particular position, if you map that you see a variant, a real frequency, fast, it's a mutation in 67% of your DNA strands, of your DNA strands. So interesting is for this particular gene, TNF-AIP3, which is a regulator of MyD88, that for this particular zinc finger protein, there's not only a mutation at that position, but that is exactly the same position. There's also deletion. So it's a lost and mutated region. So really, you see the advantage of having both copy numbers and mutation at the same time. The whole story started with Daphne, where she really wants translocations. So now we got our point mutations and copy numbers from this formalin-fixed material without a matched normal. So we want to continue on that path and see if we can also do a translocations. So to do those translocations, we basically use those same capture bottom, and we make capture probes right around those regions where you have those chromosomal breaks. So those translocations, we make capture probes right in that region. It's very much alike there where fish probes are much bigger. Those capture probes are maybe like around 100 base pairs or so. And you make those capture probes and you capture your DNA fragments. And if those DNA fragments include the translocation, you will capture also a little bit of your translocation part. And so by the time you start the sequence, you have those split reads, because I said we sequence from both sides. So at the very top, we have the split reads. We get a read from the one side, and from the other side, even the other side may be in another gene. And if that very often happens, then you say you have translocation. And you also have the discordant reads that are completely separate on the sequence reads, but every time it's the same gene or the same region. So you have basically a fusion B. So here with the capture probes, you can capture also the translocation part. It's the very first, like, let's say, 100 base pairs. So like the size of about a capture probe, that's about how long those reads are. Probes that we've used, in this case, we've made capture probes in the region of the genes that Daphne already mentioned, the BCL6, the immunoglobulin partners, the cyclin D1, and of course the MYC translocation. And particularly this MYC translocation, it's a very, very big region where you can have breakpoints. So there's not every time one exact breakpoint like the BCR-ABL or something, but particularly with MYC, you have a whole big region, up to like more than a megabase or so, where this breakpoint can take place. And as I said, every time there's different partners. So we've also tried to put probes on the partners, but very often we do not. Well, there's very unknown partners that we haven't heard about yet. So what does this look like? So basically here on the, this is an IGV view. So it's a screenshot of the broad integrated genome viewer, the IGV viewer, and on top is the ideogram of chromosome 3, and on the very right you see a tiny little red dot, and that is the actual location of which the bottom of the string is a zoom in. And so it's about 500 base pairs, so it's very, very small. 500 base pairs, and we see the base pairs from left to right. And if the base pairs match to the reference genome at that particular position, well, then you have like the gray or the orange. But if they don't match, you get another color, depending on what transition or inversion this is. And then you get the different colors like what is it, blue, red, green, purple. So here you have an example and you have an exact breakpoint where we've captured the left part. You've captured this, you get those sequence reads, and they run into something else, and that's why they don't match anymore at that particular position, because they match someone else. So this is BCL6 on chromosome 3, and maybe to an IGH part. You can tell that from this, but there's mismatches because it belongs somewhere else. So very cool here with capture enrichment. We can also get those translocations. And all 3 in one NGS workflow where we have copy number aberrations, mutations, and translocation all in one NGS workflow. And I've shown you the different examples. So here on the left, it's a little annoying to have them all separately. So the people in the lab, they have made this very beautiful circus plot. And on the circus plot, on the very outside, you see the mutations, and then the black, gray, white ring inside there are the chromosomes. So from the very top, it's chromosome 1, and then you go all the way around to chromosome X, and then on the outside, all the mutations. On the inside, you see our black little dots, which are our copy number bins, and again, with red and blue, we see the gains and the losses in there. Very pretty. And then, the cherry on top of the pudding, of course, is the translocations, and those are the black lines we see in IGH, BCL2 translocation, the black lines in the middle of the circle spot. Excuse me. And we see a MYC BCL6 translocation. We also see that at MYC and BCL6, there are some interesting gains and losses in those regions. Now, let's now pull in the fish of those actual samples. So on the left top, we see the fish break apart probe for BCL6, and then yes, for BCL6, so that is the red arrow, is the red probe here. And yeah, we see a lot because there's a lot of gain on BCL6. We also see that the other side of the break apart probe, the green part, there's also gains, but there's a lower level of gain, so we only see 3 spots there. And then for the MYC translocation part in this case, we also see a gain for MYC, and so you see 4 red dots, but the other side of the translocation part, and then we see 1 green dot together with the red dot, so that's basically the wild type chromosome is still intact, but there's only 1 of them, so we only see 1 green spot. Now, if we would only have that fish, we would never be able to interpret what's exactly happening there. We really know this very much in detail because of this combined copy number mutation analysis and the copy number together. So you see how much extra value this gives. So yeah, I'm just showing here a whole bunch of samples that we have embarked on, and basically what this graph says, so samples that we have fish for and NGS, this capture NGS, and basically what it says is always match. We don't find things that we didn't find with fish, and the other way around, we find the things with fish with NGS back, except for this one brown example. And again, there was the problem that in this particular region, we were not able to make probes, too repetitive. Yeah, so Roche wasn't able to provide probes for that particular region, so we weren't able to capture this particular MYC translocation, which the fish did, because they are much bigger probes. So yes, we do miss some things, and then, well, we were quite happy actually with all of this, like that whole combination of everything. Quite happy that we got this far, but yeah, so Syngenta is not too far away from from us. And so together with Syngenta, we started to work on those translocations. And Syngenta has this beautiful targeted locus amplification system. And it's a complete NGS-based proximity ligation system. So the proximity deal is here. So you have your FFPE block. The FFPE is deparaffinized, so you'll get rid of all the paraffin. And then you just have your cross-linked and fragmented DNA together. That fragmented DNA, you try to ligate as much as possible together. So basically you get your fragments, because of this ligation, now get much bigger with pieces that are in the vicinity, in the physical vicinity of the of your original capture region. And so after you've done this ligation, and then you do an enrichment of those regions with a capture probe, just a very same capture probes from Roche as we used before. But now, lo and behold, because you have regions that are further away from the original capture probes, and this can be up to a megabase away, then when you start to sequence, you see those sequence bumps, really, where you have really high sequence depth around your gene, with like up to a megabase away. If we look here into some more detail, on the left we have our approach, which is capture NGS, and you see with the capture probe on the bottom in what is this yellowish brownish, and then for the fusion gene, you don't read very deep, as I said, like about 100 bases into your fusion gene. Now the cute thing is with TLA is that you can sequence much further because you get pieces that are ligated together and that are in the vicinity, and so you get pieces into your fusion gene that are much further away from your capture probe. This can be really like up to a megabase. Harman can correct me later on this, but like I think you really get quite far from the original locus. So what we've done, we've made it difficult for Syngenta and TLA. So we've collected a whole set of samples that we had fished for and that we couldn't always recover with capture NGS. So this is not a representative set. We gave them particularly difficult samples and a whole lot that we couldn't do with our NGS, but we had a positive fish signal from those. And we handed those to Syngenta and told them good luck. And so what we see that most of the samples, so there's 19 samples, most of the samples, we had positive fish, we could do them with NGS and also with TLA. But the purple ones that we see here, we were not able to capture with NGS, but we did get a signal with TLA, and we got the right signal. There's one sample that was only positive for that NGS, and the reason of that was that there was a tiny little insertion, and that wasn't really a translocation, just like a tiny little insertion with Nick from chromosome X, so like hard to call it translocation. But if we look back in TLA, we could find it, but it didn't automatically call it because you don't see it in the bumps, so to say. And and then we also had additional findings with TLA that were corrected again. So I'm going to show you two of the samples from the previous slide, how that actually looks like with the NGS and how this looks like with the TLA. So on the left bottom, we see is again a chromosome representation. It's then very bottom chromosome 1 up to chromosome 20, and from chromosome from the BCL6. So we have a BCL6 MIG translocation, and it's actually an arrow. It goes from the MIG to the BCL6 in this case. Is that correct? And you see those wider bumps, and those wider bumps, if you now take the right lower panel, you see that on top, you see that gray representation. That's the sequence depth. So we have very good sequence depth for those samples. That's those bumps that we see for the TLA. And now if you look at the NGS, we don't see those sequence bumps. And actually, it's a whole region where we just don't have any sequence reads. And And the reason is that we just didn't have capture probes there. We weren't able to make capture probes there because it's very repetitive. Now, if TLA has very repetitive regions, it will still sequence outside that. You'll see that in the next slide too. So here in this next slide, sample 18, we have an IGH MYC translocation. We see again the little bumps on the chromosome plot, and then here we only see the NGS representation, and the NGS representation. Look at the very bottom, all the blue dots. It's a very repetitive region, and since we can only sequence in that tiny little region, and you get very often only half of your sequence reads or something. There were like only 2 or 3 reads that mapped in that particular region, and a lot of them will have gotten lost in the sequence trash bin, basically. And we just don't have enough evidence to call this a translocation. And here, yeah, the really cool thing is with TLA, it's clear on the left picture. It doesn't matter if there's a region where you don't have your sequence reads, because you'll have them a little bit further, because you don't really necessarily sequence the breakpoint, but you see on the bumps that there are breaks. So this is really how TLA picks those regions up, just repetitive regions, not able to make capture probes for us. So basically, yeah, let's come to the conclusion of all this, the conclusions for Daphne and me. So yes, for FFPE samples, fish in the left column, and then the capture of the NGS in the second column, and in the third column we have the TLA results. Yeah, well, fish is at this moment, yeah, we have beautiful correlation with morphology. You can actually see your cells and where the fish signal sits in which cells. But you're also dependent on a very good morphology. Whereas we've shown that, yeah, we can work with the damaged DNA and the big fragmentation, both with NGS and with capture enrichment. And it works on most necrotic samples. I mean, yeah, we've done like also quite a few samples with this, and Syngenta has also done a whole lot of samples on the TLA. And yeah, with most DNA preparations, this works. Yeah, well, the fish, of course, this is the current standard. And the nice thing with, and that's not on this slide, but the nice thing with fish also is you don't need a whole complicated computer infrastructure. You know, it's just really like doing the analysis and doing it on a microscope, and there's a whole lot of places where this can be done in small labs. You need highly skilled people, but you don't need that complicated computer infrastructure. Now, yes, the capture NGS and the TLA, they can also be standardized, of course. So yeah, we need pipelines. A lot of the pipelines are made. It takes a little longer because yeah, you have to do your DNA isolation and your cross-linking. We need all those steps, so it does take a little longer, at the moment 1 to 2 weeks before you have the results, because you also need your bioinformatics in the end. And yeah, Syngenta has this all automated already. Yeah, fish is difficult to interpret, as I said, and the capture NGS is quite easy to interpret, but you do need that IGC. You do need to manually curate, which is not the case for TLA. You just look at your bumps so you know where you are. And then finally, yeah, with fish you can do copy numbers and translocations, but not mutations. With capture NGS we have this already. We have copy numbers, translocation mutations, and for TLA, well, there's basically nothing to hold them back to also include capture probes for mutations and make genome-wide capture probes for copy numbers. And yeah, thank you very much. And with that, I will like to hand over to Harma.
So much, Bauke and Daphne, for this very nice overview. My name is Harma Feitsma, and I'm the head of Business Development Oncology at Syngenta, and I'll use the last couple of minutes to tell you a little bit more about our company and our plans. So many probably not have heard of Sargentis before. We are based in Utrecht in the Netherlands. And since we started in 2012, we have developed our proprietary TLA technology that Bauke just explained. And we provide this as service and in kits. Our business so far has mostly been applied to cell material. We, for example, analyze transgenic models and we validate production and therapeutic cell lines. But with the help of Horizon 2020 SME funding that was granted by the European Union 2 years ago, we have been able to develop the TLA technology also on FFPE material. That is what has been shown in this webinar. And that allows us to address now oncology applications as well. So the results that Bauke has presented are part of a larger study with 5 clinical sites in the Netherlands and over 100 lymphoma samples. And this study will be published very soon, and we're happy to send you a copy once it's available. We will make available as a research service an expanded version of this TLA panel, and we'll soon also release it as a kit. And for that, if you're interested to join as a testing partner in an early testing program, please contact me and we're happy to discuss. Of course, you then need to have access to NGS facility, but the bioinformatic analysis can still be done at Sargentis. And to make this product also available for diagnostic applications, we'll soon start a prospective study with 3 clinical sites. And that will be the next step on the path working towards a CE marked kit. We will also validate our TLA technology for other cancer types for the detection of translocations to enable a single NGS-based diagnostic test that can detect all types of mutations, small mutations, structural variants such as translocations, and also copy number variations, as Bauke just has explained. And we're currently starting studies on lung cancer, sarcoma, prostate cancer, and a few others with different clinical partners. And for some, it's still possible to participate. Please reach out to me if you would like to join. In general, if you're looking for a robust technology to analyze structural variation or translocations in genes of interest, we're always happy to discuss a project using TLA. So please contact me for that. My contact details are in this slide, and you can reach out to me. Thank you very much for attending this webinar, and I'm handing now back to Alison.
Great, thank you so much, Harma. As a reminder to our webinar participants, if you have any questions, please type them into the Q&A panel on the right side of the webinar presentation. We will now begin the Q&A. Our first question is for Bauke. Why don't you use RNA-seq for detection of these translocations?
Yeah, well, good question, of course. And so if you use RNA, you can only work with the transcribed RNAs, and a lot is not transcribed, first of all, and the other thing is only fusion genes that you look at then. And here we have translocations where you have, as I explained before, MYC, that is very often regions which are, for example, promoters or enhancers that are far outside the actual coding gene. So yeah, this would not work for this particular situation. Yeah.
Okay, great. The next question is for Harma. What is the amount of input DNA needed?
So first of all, the TLA protocol does not start from isolated DNA. That's important because we do the proximity ligation inside the FFPE block. So we need to start from a scroll or a section of the FFPE block. But usually, for most of the tumors that we've seen so far, just one section is sufficient to do the analysis.
The next question is also for Harma. What is the time to result of the TLA assay?
So as Bauke explained in the table also, it's around 1 to 2 weeks. And the additional time that is needed to do the TLA proximity ligation preparation, that's just 1 extra day. So if you compare it to your standard capture NGS workflow, it's 1 day extra to do the TLA preparation.
Great. The next question is for Daphne. How important is and will be detection of single nucleotide variants and copy number variants in lymphoma?
Well, that's a very interesting question, and we have a short-term and a longer-term answer to that. The short-term answer is that in practice we already use single gene mutations in lymphoma diagnostics, as pointed out, for the diagnosis of lymphoplasmacytic lymphoma. Demonstration of MID88 mutation is very important, as is CXCR4 mutation, as that impacts even on response to treatment. Also copy number alterations, for instance, amplifications as at 9p24 in Hodgkin's lymphoma and other types of lymphoma. It's the locus where PDL1 and PDL2 ligands are located, important for response to checkpoint inhibition. So that we already use. What I expect for the somewhat near future is that we will make major steps to identify meaningful mutation and copy number profiles that either impact on prognosis or even on predictive for response to treatment. And hopefully in the coming few years, trial results will emerge that show the importance of such approaches. And it is important for pathologists to be ready for when it is pertinent that we provide that information with our diagnosis as well.
Great. Our next question is for Harma. Can you also detect subclonal events, and what is the detection limit?
Yeah, so within the study that we've performed on these lymphoma samples, we have done some dilution series, and we can detect to around 5% of translocation presence, certainly. So that's well below the detection limit of fish also. And it's quite similar to the detection limit for NGS. So in that sense, we would certainly be capable to also detect subclonal events.
Great. And the next question in here is for Bauke. Can copy number variance detection also be included in the TLA assay?
Yeah, I briefly alluded on that already in my talk. So yeah, it's just a matter of putting a copy number probe. So you just put probes genome-wide, like let's say every half megabase or something. That would be one approach. We still have to deal with the fact that we like to work single channel, and another option for scientists to develop this would be to go with the off-target. So you always get a lot of sequence reads still, like maybe like 20 or 30% of your sequence reads are just like random sequence reads throughout the genome, and you can use those. We've published in the past in genome biology, where you can use those off-targets, and this would, I have no doubt that I would work for the TLA system too, because in the end, TLA is still just a capture system, so that should work for TLA as well, and get a copy number from the off-target. So there's two paths they can take. Yeah, I'm curious how it's going to pan out. It's important to keep in mind that, yeah, I think we would still like to work without a reference signal.
And our last question is for Daphne. Why can't you assess MYC translocation with immunohistochemistry?
Could you please repeat the question? I couldn't hear it properly.
Sure. Why can't you assess MYC translocation with immunohistochemistry?
All right. Well, the point with MYC is, and that this was also briefly alluded to when Bauke answered on why we cannot use RNA assays to identify MYC translocation is that the breakpoints are located around MYC, leading to overexpression of the MYC gene, but also MYC is regulated by different mechanisms other than translocation. And these events all lead to overexpression of MYC, meaning that overexpression does not equate translocation in the case of MYC, as is actually the case in, for instance, BCL2 or BCL6 translocation. So that is not suitable in this case.
Okay, well, that's all the questions we have for today. Let me thank our speakers, Dr. Daphne de Jong, Dr. Bauke Ylstra, and Dr. Harma Fietsma, and our sponsor, Sargentis. Again, if you submitted any questions that we did not get to, we will have our experts follow up with you afterward. As a reminder, please take a look out for our survey after you log out to provide us with your feedback. Thank you for attending this Genome webinar.